I just don't see it.
So line 37, unknown white sponge, it doesn't have a smooth edge the way this one does. So the only one that kind of looks like this is line 30, unidentified demo sponge, I think. But it looks a little more clean and almost like it has an undulation in it versus this kind of real flat.
This one's definitely
Meredith's saying she doesn't think this
sponge.
one's on the list.
Meredith, do you want us to?
We already have samples of this. No.
Okay.
Say we already have samples of this.
Okay, continue on.
Pretty though.
Okay.
So we'll continue on?
Yes, thank you.
But you're right, Steve. I mean, I've been surprised how few sponges we've been encountering so far.
Oh, it's just a little anemone.
There's a small chitin on the rock there.
Oh, yeah. So circling back on the predatory sponges, Abigail gave some nice information here. So instead of being filter feeders like most sponges, the predatory sponges have these hook-like spicules. So the spicules kind of help make the skeleton of a sponge. And their spicules are hook-like so that they can capture their food with that. And then she said the ones that look like pipe cleaners, like we saw just a moment ago, are in the family Cladorhizidae, and the genus is Abestipluma. So that's where I was getting
That's okay, Bob. I was just making a note of it. We don't have to get close imagery.
What is that? Is that a tiny coral?
Yeah, it looks like a small
Yeah, it looks like a
swift,
small chitin.
or something.
Want to zoom in again?
Oh, that. Huh?
Okay.
Ship move
A
has
little
started.
sponge behind it.
We gotta get going.
Okay.
Zoom
So
out.
there's one of those predatory sponges in the upper left as well, if people just want to look at that again. We don't have to zoom on it. I was just pointing it out.
Viewers at home can replay.
I gotta get going.
Yeah. No, but it's fun. So we, you know, as we go along, right, we see all these larger animals. And then when we zoom in, we see that the rocks really are just covered with so much more that we can't see from the distance.
Yeah, let's continue to keep exploring.
And, you know, Jan, I think that speaks to some of the reasons why eDNA is so exciting to think about, because all the animals that we see, whether they be small or large, are all shedding DNA into the water, just like we lose hair and, you know, we slough off our skin as we go through our daily life and leave little remnant DNA fragments of ourselves behind. The animals do the same thing. And so if you get all that in a water sample and you can look at it, you'll be able to see more animals than you can see with your eyes from these camera views.
Zoom in now.
So the palm frond version of Asbestipluma last year, there was a lot of krill in the water, and you could see the little kind of hooks or Velcro, just kind of capturing and having those the little krill or amphipods, whatever was in the sponge, and inside the sponge you could see little inverts in there. It's very, very cool.
That's true. Nice little, we've got a Dover sole and coral. The top of that coral is missing. It's polyps.
Yeah.
Wonder if that's actually a Lepidopsis bamboo coral, if it just hasn't branched yet. It's sometimes difficult to tell. Looks like a lot of these corals around here, the bamboos are branching, but not until it gets higher up in the colony. This one looks like an Isidella.
So this one has those, the sweeper tentacles that help us to know it's an Isidella, right? What was right below it on the rock?
Got it. Okay, we gotta get going though. Zoom out.
Yeah.
If we do want to stop here and take a look around, I can hold the ship.
Okay. That'd be good. Thank you.
Would you like to do that right now?
Yes.
Bridge nav.
Jan, is there any chance this sediment is thick enough for a sample?
think
Whoa,
so.
look!
There's
Did you
not.
see
That's right on
flapjack
top.
to the left?
Oh. It's off of my screen.
Good eye.
Oh.
It's
Ha!
quite
First octopus
impressive.
sighting. Nice.
Wow.
All right, Al.
That's a big flapjack.
Zoom in.
Yeah, let's make sure we get the lasers close, but then we can take the lasers off.
Okay, lasers off.
Got it.
Hi there, buddy. I can only imagine how it feels to have these bright lights shining on you when you're used to living in the dark.
Do we know what the 2 rows of dots
And-
are at the front of its face?
I do not, though it looks like they continue down
Oh.
the sides as well.
Yeah.
Whoa. But let's see if anyone online knows anything more about flapjack, and I can look online too and see if I can find anything.
I know there are a couple of different species that are very similar looking. And flapjack and Dumbo are often interchangeable, but curious which one this is, if we have any cephalopod folks watching.
Is that good? Got it.
Yeah, thank you.
That's
Zoom out.
a image.
It looks like its left eye might be a little either closed or not sure.
You want to zoom again?
Well,
What's happening?
I'm just the
on it,
navigator.
Probably. I don't want to harass him too.
Yeah.
All right, well, the ship is stopped, so if you want to look around at anything else, now's the time, or we can keep moving.
Well, Jan, what do you
is
think about the sediments here?
No, I don't think so. I mean, we could try.
You want to zoom in on that?
I was just curious to know how thick they might be.
Mm-hmm.
How thick do
Yeah,
the
like-
sediments need to be for them to stay in the core?
It depends on how granular it is, you know. If it's like, I don't know, coarse sand, it might fall out of there pretty easy.
So what do we think this is?
Okay, zoom out. Oh, are you still looking at it? Zoom in again.
Is it a coral? Looks like a coral.
So I was thinking it was on the right next to the mushroom coral on the list.
Gersemia. Simia. Gersemia.
But it had already been collected.
Mm-hmm.
Steve, do you think that's Gersemia?
Yeah,
Or
that's my
Alcine.
understanding as well.
Yes. Meredith says yes, Gersemia.
Right.
Do you want to look at anything else around here? He's just that
Yeah.
octopus is just hunkered down. I don't think he's gonna do anything else for us.
Hey, Steve, that was a number 4 video highlight.
I did mark a video for highlight.
Excellent.
Yes, I'm slowly adapting.
I'm glad that impressed even you, or you knew that it would impress us.
I was gonna go 3, but I was like, no, you know, I'm gonna.
There was some excitement there.
That's our first octopus, I think, right?
For each
That
one.
I
It was,
know.
yeah.
Oh, you guys,
not
can you
far
see
as I
that
know.
little deep sea sole in the bottom left going out of screen? When they're that little, and they have this chocolate colored background, and then the coloration on them is like light blue.
There it is, right there.
They're really beautiful. Can you
Zoom
see
in
that
now.
little bluish, sky blue tinge to it? Oh, he's coming out, or she's coming out for us
Oh,
to take a
wow.
look. And then the underside, if you're able to flip it over, the underside is chocolate brown, which I've never seen on another flatfish. They're really cool.
It's interesting. Usually their undersides are a lot lighter in color.
Yeah, usually they don't waste the pigments on the
Yeah.
underside because you don't see it. I was really surprised when I saw that.
Do they spend more time sort of hovering above the seafloor, so they're perhaps trying to just blend in?
Not that we've seen.
Yeah.
That, you know, what do they do when we're not looking? I don't know.
That's the question here for everything, isn't it?
Okay.
The $50 million question, Samantha.
Yeah.
What do they do when we're not looking?
That's why drop cams and stationary cams are important for long-term observation. But then they know
Yeah,
that the camera's
eDNA
there,
won't help
so.
us with that.
Yeah.
It reminds me of the Gary Larson cartoon where the cows are standing up and they're talking, and then then one calls out car, and they all stand in their field
Yeah.
as the car drives by.
Exactly.
Car leaves, and they all go back to their upright positions, talking, having their coffee break. Yeah, Guy is saying that he just thinks this is a pretty thin veneer of sediment here. We're not going to really be able to get much.
Do we want to keep looking around here or continue along towards 2S?
Let's just continue along this contour.
Okay, copy that.
And then when we get to our little, when we get there, we'll reassess, maybe go up a contour. Yeah, we're about halfway through the entire transect, and let's see, we have to start coming up at about 10:30 at the earliest, maybe 11 at the latest.
Wait.
So we have plenty of time.
Oh, okay, wait. You're talking about tonight? 12
Tonight.
hours. Yeah, okay.
Yes.
Okay, got it. Seems like the thorny heads really like this habitat, as well as the flatfish. Little nooks and crannies to tuck in. They're not out in that little flat part in the middle here. They're all finding a little somewhere to tuck in. Little hermit crab is at the bottom of the screen there. So Jan, yesterday I think you were saying that this part of Pioneer Canyon historically has more fishing in it than the part we were in yesterday. Is that right?
It's around this area. So we kind of took a big kind of cross section of it. And this might be a soft sediment area.
Would you like to stop the ship to take a sample?
Yeah. Let's stop here if we can.
Can we get the lasers near that huge fish on the right, the big thorny head? Just curious to see.
Trying to do a tube core? Good. Let's see.
I think they're evaluating that right now. Science, is this a good spot to do a push core?
Maybe.
I don't know if it's a good spot, but I think we should try.
Copy that.
Seems to be a pretty large thorny head there.
I know, right?
Do they have
Being
any
harassed by a hagfish.
That's what I was going to ask. Is there any association there or not known?
I wonder if the hagfish is trying to avoid our lights.
Nice little hagfish scarf there.
These thorny
So I
heads do
think
not
for,
seem that bothered by much.
No, they don't. So I think it's a short spine. Thank you, Caitlin. I think the area right behind the ROV at this point had some bioturbation in it. And that's why I was thinking that maybe there's enough soft sediment there for a push core. Could be wrong.
Oh, you wanted to go behind us where we're at?
Yeah, if we can.
Might have stirred it up.
If not, we can just keep moving in this direction. Well, we did find one thing that bothers thorny heads. That's it.
Okay, where are we thinking here? Anywhere here?
Right behind. Yeah, right behind where we were. Yeah, in this area.
Like where the lasers are, or seems
where the track marks are from the ROV. Yeah.
Oh,
in this
well, that's all
area.
disturbed now. Is that okay?
No. That kind of area. I guess we could we can push on.
I mean, it's like.
I think
Well, what about
that
just
area where
to the
lasers,
right of our?
Wait, the right of?
Well, I was thinking if you look where the ROV was, and then we just kind of go to the right of that, enough that hopefully it wasn't too disturbed. There's a lot of bioturbation there still, Jan.
Like where the lasers are now?
Further down?
More to the right of that tiny thorny head is what I was thinking.
Like,
Here, let me draw it up there. In this area.
I think the
when
flatfish
you're in
is
the van,
pointing.
you get to point. Yeah, I was
Okay.
thinking in front of the flatfish.
Or
Yeah.
this area generally.
Use.
Yeah.
Look out, Mr. Flatfish.
Yeah. In there.
I don't know.
Nice, Steve.
We can try.
Excuse us.
Yeah, right in there, where the lasers are now.
Okay.
Gonna have to wake up that flatfish though.
We're a little nose down, so he's probably okay back there. I'm gonna turn on the porch light.
Yeah.
Okay. You want to switch over to sampling mode?
Yeah.
And then we need to switch the camera to, I hate that I'm up there. I hate that.
Starboard buy box.
Just the nav.
Okay, you want to come out?
Now you know what we see when we watch you drive.
Yeah, I know.
Oh yeah. How far?
Out more. That's good.
Okay.
Group force. All right.
So Jan, what are we going to use this sample for? If we can get it, that is.
Let's see. Can you switch back to dive mode?
Yep.
Jan, are you there?
And
Sorry, I was
Just see
drinking
it on
coffee.
bubble now.
Oh, sorry to interrupt your coffee.
Yeah, so the group at Bodega Marine Lab studying ocean acidification and ocean
it's very
climate
easy to get up in
and
the
climate
light bar with
impacts.
it. The coral cutters.
And also looking at just the growth and health of calcifying corals, and really looking at that top layer to see what they're feeding on.
Don't have a great grip on it, but where we're at here.
Any area in
That looks
particular?
pretty good to me.
Yeah.
Go for it.
We'll see how deep this sediment really is.
Cool. Okay.
Is that about it, huh?
Well, I just don't want to. If I'm hooked into it
Yeah.
and the vehicle starts moving, that's bad scene.
Oh.
Oh, that's a little further.
Pretty hard, but
Yeah. Be great though if we get some of that harder pack at the bottom. That might help hold it in the core, because the top looks very loose.
Yeah.
oh, the ball's stuck on there. Can you zoom in on that now? Huh. The ball got stuck in the fingers there.
Yeah.
That's one way to pick it up.
You're gonna be stuck again?
Yeah.
Probably. Yeah. That's kind of a bummer.
Guess we need a different shape handles.
You're not one of those hockey puck people, are you?
I didn't say hockey puck. I was very neutral in the words I used.
I don't know if we're gonna get much more than that. Is that okay?
Let's see what happens, right?
Yeah. Yeah. Hopefully it won't all fall out. Yeah, I think that's deep enough.
Yeah, it's not very impressive. It looks like it's going to fall out before I get it over there. Yeah,
Okay.
it's no good.
Yep.
You could try
Well, can't
somewhere
hurt to
else.
try, right?
Yeah. I just don't think this is a good area. It's got a lot of kind of fresh slump going across it.
So we're going to try it again? Or,
No, I don't think so.
okay.
Not right here.
Putting it away? Putting it away.
Yeah.
Back to
Yes,
simple.
please.
Pay attention. Out of the way. Got the wrist drift.
All right.
Hang on.
Take it in.
Right. Yep.
All the way?
Yeah.
All right. Also, before we take off, take a look at the rear cam on Herc. That's mucus. Oh, okay.
Yeah.
All right.
Not, that would be bad if that was the daisy chain.
Yeah.
A lot of
Schmutz.
Herc's mucusing.
So actually, I need to run off to the facility real quick.
Quick bio break. Science, our pilot's taking a quick bio break. Do we want to plan out our next move after this temporary waypoint or temporary target that I dropped here? Do we want to continue down this contour line east and then southeast, or do we want to head into the kind of the bottom of the unknown area here?
I think what we want to do is go to your temporary waypoint and then go up one contour, take a look around, and decide from there.
Copy that. Okay, so I'm going to put another temporary point here. Does that look about right? What you're thinking?
I don't see it.
I
don't see it yet.
Take a second. I think we got about a 40 second delay.
There it is. Do you see it, Jan?
I do not.
Maybe
we have a longer delay.
I think Jan and I have different delays, speeds.
Yeah, let me see if there's a way I can refresh, because I just went out of HiPAC and I came back in. Can you move your cursor at all?
Sure.
Sam?
I wonder if we can put HiPAC on the telestrator.
That would be cool. But
there we go. Now I can see your cursor moving.
Okay.
But I don't know if Jan can yet.
Gotcha.
Hold on a
Yeah, we've
second.
definitely
I'm
found
getting it back up.
home Wi-Fi speeds to influence as well the delay.
I closed out a few other windows too.
Yeah, I moved my whole setup to a different location closer to the router so
There
I
it
could
is.
Ethernet in, and I think that definitely has helped
Yeah.
me have a little bit less delay.
Yeah.
So Sam, I would think that I would move that target directly above or north of the current temporary
Gotcha.
target. Yes, right there.
Okay.
Yeah. So that was quick. I can see that now. I just had to go out and come back in.
Perfect. Okay, so pop up there, take a look around, and then decide our next move. Perfect. And how much time do we have to get to 2S?
We have plenty of time.
Okay. Because right now we've got...
Let's see, what time is it? We
830
have a
meters.
little, yeah, we have a little over 12 hours to get to 2x, the end.
2x, the end.
The end of our, yeah.
Great.
And this seems to be the waypoints with the largest distance between them.
Perfect. Okay.
We could zoom in and just see what's in the rock that's directly in front of us, I guess. Or can we not zoom while he's not, well, he's gone. I don't know I know there's a dance between Herc and video, but I don't totally understand who does what, so.
Alrighty. We have Herc pilot back in the seat, and we can continue our move here. So I will, our next ship move, bridge nav. 50 meters, 080.
What are we doing now?
We're heading 080.
But I mean, in general. What are we transiting?
Oh, we're just transiting to a next spot where we're gonna, do we want to stop at the next target here, or do we want to just continue on to the next contour line?
Let's take a look and see. We're in explore mode.
Roger.
So
explore mode.
I think it's whatever we feel like doing.
So I have been doing a little looking around about the octopus that we saw, and I have to say the one that we saw resembles a lot of the photographs that I see for what are being called flapjack octopus, or Ophthotuthis is the genus. But I haven't seen anyone talk about those kind of dots that we saw down the mantle of the octopus and what they're for.
I can't recall if I've seen those on the
So this current is pretty much head on, right?
flapjacks.
Yeah.
But I'm still looking, so we'll see. Maybe I'll find something.
Get down closer.
Yeah, working on it. Yeah.
But this type of octopus is different from maybe the more typical octopus by having that the mantle extends down the arms and creates like a web. So when they're swimming, they look more like an umbrella.
So Jen, you're breaking up a bit to me. I don't know if that's the same on your end or if anybody else is hearing that, but
Loud and clear for both of you from the ship.
okay. I think I'm going to have to get off of high pack if that continues. High pack on my computer that I have the V-Link going through. It seems like if I have just too many things going on with V-Link on my one laptop, it starts wigging out.
Well, Jan, if you want to keep HiPAC on like you did the other day, and I can convey to you information from the science chat, that's possible. Whatever works best for you.
Yeah, it's just that's what we would end up having to do, because then I run HiPAC and the science chat through the same computer, and V-Link is just the only thing that's open and on my other computer. We have high tech going on downstairs in the garage. John's workbench.
Can't hear any. If it's a noisy work, we can't hear it.
No, it's all electronics.
Oh, got it. I mean, that was another stretch of soft sediment, but I'm assuming it won't be much better than what we just tried to core.
I don't think so. I think there's some more soft up ahead.
Okay.
A couple of, October 8th, World Octopus Day.
Huh. Wait, is that today?
That's
I don't even know what day it is. You know.
I have to look at computer.
I'm sitting in a darkened room. I have curtains on the windows. It's a little cave. Oh, there we go. It is the 8th. Nice. So that was just the first of many, hopefully, octopus to come on World Octopus Day.
Yeah.
Maybe take a look at that white thing.
I have to say that the number of cephalopod holidays online has greatly increased. There's now a couple of weeks. There's one in the summer as well, which is great. You can always use more cephalopod awareness days, but there's quite a number of independently organized celebrations.
All right, Al, can you zoom in a little bit?
All right, Al, can you zoom in?
But here on Nautilus Live, every day is Octopus Day.
That's also true.
Isn't that a slogan?
Yeah,
I don't know about
so.
yesterday.
I think we say that every time there's
yesterday
an
we were
holiday
kind of
about some sort of ocean animal.
Well, yesterday, I feel like yesterday was Gastropod Day.
Officially or on
with all
Nautilus
the
Live?
egg towers.
Oh, okay.
I think it was Nautilus Live, Gastropod.
Right,
Especially Egg Tower Day.
Yeah.
I mean, there's that one. I don't know if you all saw it. It was in the what time? Maybe it was Eric Burton's watch. So that's 4 to 8, and they came across just this field of snails and egg towers, but it wasn't the yellow egg tower we'd been seeing earlier in the day. It was white. And it was a whole city. I mean, there was like 3-dimensional structure. There was like skyscrapers and condominiums and then, areas where it was just snails gathered in these big, it was really cool. I got a few screen grabs I can send to you, Jan. It was really neat.
Yeah, I missed it.
We were calling it me and my kids were calling it Snail City.
Oh, Snail City. What's the video game that you can build a city?
Sim City?
Sim City? Is that what it is?
Yeah.
Maybe that's what they were playing a video game.
I don't know. Gastropod City sounds just fine to me.
Yeah. I was going to say this is all kind of a simulation. We're always kind of playing a video game up here. Or pilots are.
Yeah, I mean, when you see the controllers, it really does look like a very fun and detailed kind of game console for a video game. Is that a little kelp to the left?
Oh.
You want to stop for it?
Which one?
Sure.
Bottom
That's
left.
up to Jan.
Yeah.
Yes.
Bridge nav.
Do we have an interest in a specimen of a kelp?
What are we looking at?
Pole position.
Kelp? I think this is what they're looking at in the telestrator. But I don't know if
So it was, yeah,
It's to the left
the
of that thorny head.
Is it kelp? What is it?
No, it's just below the lasers.
kelp frond, I think.
yeah,
Can you just zoom in and
zoom on
see?
That's what we're interested in? All right, Al, can you zoom in a little bit?
It is kelp.
So Jen, why is
All right, let's zoom back out.
kelp on the C4
Well,
special?
Is that something that
Yesterday I heard Taylor Ann mention she might want another sample or two, only if it's convenient, she said, because
Of
she's
the kelp?
looking at microplastics that, but we don't have to stop if we want to keep making
Well,
time to our next point.
We're stopped, right?
Where would you put it?
You want to
Wanna
park
go-
it there? Park it
Yeah.
in front, so
It would have
pan
to go in
down
a
some.
slurp because otherwise it would just float out of a bio
You can
box.
see the front of the box.
Yeah, I agree.
Yeah.
And then, yeah.
Park
Set
it
it
right
down,
here.
So that
set
seems
it down
wider.
gently.
Oh.
Well, a little bit
Oh.
further forward. Right there. And come in easy. And
Do you want to try kelp slurp, Bob, number 2?
Are we
or-
close enough?
that's a little bit far, but it's better
This
if you
seems
were
though
to
like
get over
wider
that way,
than
so
what was
keep the sampling like
slurped
right
yesterday.
in there. That's perfect right there. Plant that.
All right. Oh God, the current.
Yeah. So now when you need to give it some forward stick and down stick, like to hold it stable. And then when you're all stable, then you hit stick lock. You good?
Yeah.
Are you pushing forward? You're not pushing forward. You got to hold it steady, like forward and
Down.
well, don't put lateral. Just drive into the slope. So down and ahead just a little bit, and then when you're there, hit stick lock and let go of it.
All right.
And then the sample comes to you. It's perfect.
It wants to be collected.
Yeah, don't go anywhere.
Uh-oh.
We got a tumble
All
I-
cart.
right,
We
what
may have spoke too
chamber
soon.
is this going in?
Whatever you got, they're all open.
Uh-oh.
Might have to.
We got a runner. A drifter.
I thought there was a piece of kelp in one of the slurps.
It's in the flush. We don't typically
no.
Oh,
call
it's
it
in
a
the flush.
sample, but
Oh.
I've made a
Right,
note of it.
Oh,
Uh-oh.
uh-oh. Maybe not.
Bob's gonna chase it.
Maybe.
That kelp is smarter than most of the things we sampled.
Yeah, where'd it go?
Wow. It's gone. It got out of here.
Yeah. Well, that's the one that got away.
Tumble.
Okay.
Is that the kelp on the right over
Oh.
there? Or is that a fish?
That's a fish.
Oh my goodness.
I don't know what happened. The kelp was gone.
Okay, that's fine.
That's a new one.
Kelp's
Wow.
pretty tricky stuff, it turns out.
It's outwitted us again.
The wily kelp.
First I got stuck in the slurp for over.
Yeah, and then it
12
shows up
hours.
magically.
Yeah.
Yeah, in a follow-on interview, what's the hardest thing to sample? Turns out it's kelp.
Kelp.
Well,
Okay,
I
well,
know.
do we want to look around here a little more or continue on to the next temporary waypoint?
Let's go ahead and crab along this, go sideways along this contour
Yeah.
Okay,
to the east.
okay, so
Heading east.
scrap the waypoint right above us, north of us.
Oh, I thought we were at it. I'm sorry. I took a really quick look. Yeah, let's continue on to this temporary waypoint.
Okay, affirmative. Bridge nav. Three 0 meters, 008. .008 is your new heading. Pilots.
Roger.
Steve, could you log that kelp attempt as a, I don't know, 3 maybe? It's pretty good.
I'll do that for you.
For a
Sure.
Yeah, well,
maybe a sampling reel?
attempted kelp
Foiled by
sample,
kelp.
but it got away.
Yeah. I think it's important to show the wide range of how sampling actually works or does not. Gotta have some fun moments.
Well, it taunted us too because it looked like it was just lining itself up for easy sampling, and then it was like, psych.
See ya. I mean, to be fair, we are a deep sea research group, and kelp is a little shallower, so we just don't know the behavior of these strange shallow water creatures.
There's a fish over there.
Let's go find out. I can lateral over.
What's that round shiny thing? Oh, blob sculpin on the left side. Upper left. Upper left.
The large fish?
White thing?
I think it's, oh no, sorry. It's a thorny head.
What's the white thing between the two fish up
It's
there?
a big thorny head. Wow.
A white thing between, oh, like this square thing looking? The
Yeah.
square looking thing?
It's the side of an anemone. Or like looking at it on the side. Body wall.
Oh, yeah.
All right, Al,
Nothing
can
to
we...
see here.
All right.
And Herc, the ship is heading north, and you're still
North.
headed east.
North.
What'd you say the heading was?
008.
Oh, I thought it was 080. That'll
Used
do
to
it.
be. So we need to zip. Gotta put the pedal to the metal, turn it around, and take off. Well, follow the nav. So you need to go westish. How's our wraps? Yeah, we kind of need to do a full
Oh, okay. The other way?
spin. Just come up a little bit. Well, we're kind of stretched out. So just give it full tilt sideways.
All right,
Yeah.
full lateral.
Show the ship for a minute.
We'll let them give it a whirl.
Okay, literally.
We're not too far out of whack here.
No, Tom was saying that the
All right.
large
Now you
thorny
can
head
turn
is a
ahead
short
and I'll
spine,
turn Argos.
and the short spine thorny heads get twice as large as the long spine.
So you can look in the overhead view and you need to zip ahead.
Yeah.
Okay. That's good.
Okay, we are just a couple meters away from our, 1 meter away from our temporary waypoint here. So do we want to stop and take a look around or continue down the contour?
Let's continue east contour.
Roger, east on the
And
contour.
Are we zoomed all the way out? It was just the slopes. We're just more in the slope. Oh, okay.
Bridge nav. 40 meters, 090.
Will you go back onto the Hercules on yours?
Oh, sorry.
No worries.
Pilots, we're heading now due east.
Back to 090.
090.
Okay.
We reached our other waypoint.
You okay on me switching?
Yeah, yeah.
Get over there. A little at a time here.
I'll lateral over a little bit and keep pushing ahead.
Is that on the left-hand side?
What's that white thing?
Is that a sponge? You think?
Trying to get back over there.
Want me to handle this?
Yeah,
Right
sure.
ahead.
Happened to the light.
You want to plant it right there?
And we have time for a quick zoom here, but if we want to
Yeah,
stop any longer, we'll need to stop the ship.
Go for a zoom.
Looks like a
Yeah, I
similar.
don't think we need to. Yeah, to what we saw earlier.
Yeah, Posolastra.
Okay.
Sponge.
So Sam, Jan stepped away for a moment. When she gets back, maybe we should talk about whether or not we'd want to do a transect if we keep seeing this relatively soft area. We don't have any soft
Alright,
Alright, good
areas like
captures.
this, so.
Roger.
So one of the things I miss not being on board
Thank
the ship
you.
this year is
This is
the,
still only my 4th time piloting, I think, so.
you're doing a great job.
Thank you.
I miss the interactions with the classrooms we would do from the ship. And that's always been fun. So I have the opportunity to zoom into a 5th grade classroom at one of the local schools today, my son's classroom. So I'm excited. I got a screenshot of that octopus earlier that I'm going to throw in there so that I can wish them all a happy World Octopus Day.
Lovely.
I wonder who we've got coming along here. Another thorny head.
A short spine.
What's the difference
You don't
between?
usually see them swimming.
Short and long spine?
Yeah. Well, I mean, I can make a guess, but is it the kind of difference that the average person can identify, or is this a...
Was it a third spine?
Is it
What
third
was it?
spine?
Third spine in the dorsal
Wow.
fin, yes. But I also think, based on what Tom said, that that size of a thorny head is going to be a short spine. But yes, the third dorsal spine is longer in long spines.
Okay. But also the short spines get larger than the long. Okay.
And another thing we've learned is the really little ones that we see sometimes, they don't have this bright kind of salmon pink coloration, but actually a darker blackish gray coloration that's only present in these smaller fish.
Oh.
Okay, looks like we're going to have Justin Hall from Greater Fairlawn Sanctuary joining us in just a moment. And I had to reheat my coffee. Oh my gosh, it is foggy cold today. Jen, how's the temperature down where you live?
So I'm in the Eastern Bay area where it can get pretty hot. We've had a number of days where it's plus 100, but we're finally cooling off, and I think it's only supposed to get in the 70s today. But it is sunny.
Get out there.
Yeah.
I'm in the coastal fog. I'm up in the redwoods, so fog is feeding our trees. They were very, very sad for like the last 4 weeks between all the fires and the ash and the heat.
So Jan, for a little while there, I wasn't seeing any rock, and I was thinking we might be able to do a soft transect, but looks like we still have enough
Is that fishing
rock
gear
here that
there?
Might be.
I think that's seagrass, but we should look. Just
Awfully
make sure.
long.
It's not. Yeah.
Get in there to see
Sure.
that, but
Looks like it kind of wraps.
Yeah, sorry.
it looks like fishing gear to me. It's like, yeah, it's on the
Yeah.
bottom.
Yeah.
Don't back up too much.
Okay.
I don't know what's going on with it.
Okay.
Hopefully it's just some line on the
Right?
seafloor.
Yeah, it looks like it. Yeah, it loops up there. Comes back down.
So Jan, I heard,
Yeah,
or I
it's all
saw
over
in
here.
a.
So
Oh, yeah.
Oh, yeah.
there's net,
You sh
right?
Yeah.
No, well that doesn't. That looks organic-y there.
Yeah.
Can you,
Nothing coming up in any other cameras.
anyway, keep your eyes peeled for.
Jan, I saw you mention in one of our kind of report summaries yesterday they saw some ordnance. Did you see it? I'm curious to know what it looked like. And is that something we should keep our eyes out for on this dive as well, or that was just something that for that section of Pioneer Canyon?
And I'm sorry, what you cut out there? What were you referring to?
In a
Yesterday?
summary report from yesterday's dive, you noted
Yeah.
that they saw
Should I just slow
ordnance.
down a little bit?
Yeah.
But I didn't see that,
Just
and I
stop
was
and-
curious
I mean, if you get out in front like that, maybe you could find something and like
what it looked like.
tell Al to
Take a
zoom